get to go through pyruvate decarboxylase / Krebs cycle. So the first thing that happens here is pyruvate is transported into the mitochondria. So remember, this is in the cytoplasm. So we have to take this and bring it into the mitochondria. Okay? So then we have our 3-carbon pyruvate, and we decarboxylate. So when we decarboxylate, we end up with a 2-carbon thing, because we've lost a carbon. We've decarboxylated. And so we've lost a carbon in the form of CO2, right? And so remember, this is a completely oxidized carbon.
Yeah.
So we've totally oxidized this carbon. So when something gets oxidized, something else gets reduced.
Yeah.
So this carbon got oxidized, so then we create another NADH. So every time a CO2 leaves, we make an NADH.
Okay.
Carbon gets oxidized, NAD gets reduced to NADH.
Okay.
So now we have a 2-carbon thing, and in biology, 2-carbon things are called acetyl.
Yeah.
So we stick this 2-carbon acetyl thing, so let's draw it. So we have an oxygen double bonded to a carbon and then another carbon. So this is our 2-carbon thing. And then we stick this onto this other huge thing over here that we don't really care about. But we call it S-CoA.
Okay.
So this is just a sulfur, and the CoA is coenzyme A. So we have this 2-carbon thing, and it's stuck onto this big CoA. Now this thing, this acetyl CoA, is what feeds into the Krebs cycle. So the Krebs cycle is sort of already going. We already have some Krebs cycle intermediates. So the Krebs cycle starts with a 4-carbon thing.
Yeah.
So we have a 4-carbon thing already. And then we mix in this 2-carbon thing, acetyl CoA, and 2 plus 4 is 6. So we get a 6-carbon thing. This 6-carbon thing is the key thing.
Okay.
So this is called citrate. A better name for it is tricarboxylic acid. Tricarboxylic acid. And so you see there's a T, a C, and an A. Tricarboxylic acid. And so some people, like molecular biologists, biochemists, they call the Krebs cycle the TCA cycle. And it's because of this thing, because this thing is so important, this tricarboxylic acid. And so that really makes the most sense. Because what's happening is we create this thing, these 3 carboxylic acids, and then we decarboxylate them, and we create these electron carriers.
Yeah.
So that's what we do. So then this 6-carbon thing decarboxylates, becomes a 5-carbon thing, and it decarboxylates again, and becomes a 4-carbon thing, and that goes through some other reactions to come back to this 4-carbon thing. So again, so remember, these decarboxylations, we're losing CO2, CO2 is coming out, and every time we lose a CO2, we make an NADH.
So every time you lose a CO2, you also make the NADH?
Yes.
Right? Okay.
Yeah. So we decarboxylate 2 of these in the very beginning of this cycle. We just goes from a 6 carbon thing, decarboxylate, NADH, NADH. Then we do a couple more steps to get this 4 carbon thing back to this 4 carbon thing. In that process, we also make a couple of different things. We make another NADH. We make a weird thing called FADH2, which is similar. And then we make an ATP equivalent called GTP. And remember, per glucose, we have 2 of these 3 carbon things, right? Because we started with a 6 carbon, we broke it into 2 of these 3 carbon things.
Yeah.
So we're getting 2 acetyl-CoAs per glucose. So we go 2 cycles of this.
2 cycles. Yeah.
So per glucose, we're getting 2 NADH here, 2 NADH here, 2 NADH here, 2 FADH2, and 2 GTP.
Okay, so everything doubles.
Right, everything doubles, exactly. So we get 2 cycles per glucose metabolized.
Yeah.
All right. Then we move on to the electron transport chain.